Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Alternative splicing, mechanism, types, and role in gene expression regulation. Table of contents. Introduction. What is alternative splicing? Mechanism of alternative splicing. Types of alternative splicing. Alternative splicing in eukaryotes. Alternative splicing in prokaryotes. Examples of alternative splicing. Importance of alternative splicing. Introduction. Alternative splicing is a regulated process in which a single gene can produce multiple proteins by joining or skipping exons in various combinations. In humans, alternative splicing increases the diversity of proteins encoded by the genome, contributing to 95% of multi-exonic genes. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. What is alternative splicing? Alternative splicing is a natural process in which the exons of a single precursor mRNA are joined in different arrangements to produce two or more distinct variants of mature mRNAs. This mechanism enhances proteome diversity and allows regulation of gene expression after transcription. Mechanism of alternative splicing. The pre-mRNA includes multiple introns along with exons. During splicing, specific exons are selected to be retained in the mature mRNA. Splice site selection is regulated by splicing activator and repressor proteins. Regulatory elements within the pre-mRNA, such as exonic splicing enhancers and exonic splicing silencers, also influence splice site selection. Consensus sequences help define important regions within nuclear introns. A GU sequence marks the 5' end of each intron. Near the 3' end of the intron lies a branch site, which always includes an adenine. The canonical or consensus sequence around the branch point varies, but it typically includes a polypyrimidine tract, followed by an AG dinucleotide at the 3' end. The spliceosome, a complex of RNA and proteins, facilitates mRNA splicing and includes small nuclear ribonucleoproteins, U1, U2, U4, U5, and U6. U3 is not involved in mRNA splicing. U1 small nuclear ribonucleoprotein binds to the 5' GU splice site of the intron. U2 small nuclear ribonucleoprotein binds to the branch point adenine, aided by U2 auxiliary factors. This forms the spliceosome A complex. Formation of the A complex is critical in determining which intron and exon ends are selected for splicing. U4, U5, and U6 small nuclear ribonucleoproteins join to form the tri-small nuclear ribonucleoprotein complex. U6 replaces U1 in binding, while U1 and U4 are released to allow the spliceosome to perform two transesterification reactions. First, the intron is cleaved at the 5' splice site and joined to the branch point A through a 2' 5' phosphodiester bond. Second, the 3' splice site is cleaved, and the two exons, upstream and downstream, are joined together by a standard phosphodiester bond. Types of alternative splicing. The mechanism of alternative splicing can occur in several distinct forms. Constitutive splicing. Introns are spliced out, and exons are joined together in a fixed arrangement. mutually exclusive exons. Only one of two possible exons is included in the mature mRNA after splicing. Exon skipping or cassette. Exon. Specific exons may either be included in or excluded from the mature mRNA. This is the most common form of alternative splicing, 30%, observed in both vertebrates and invertebrates. Alternative 3' splice site. Exons are joined using an alternate 3' splice site, resulting in a variation in the length of the upstream exon. Alternative 5' splice site. Exons are joined using an alternate 5' splice site, affecting the length of the downstream exon. Intron retention. An intron may either be retained in the mature mRNA or removed. This pattern is most commonly observed in lower metazoans. Alternative splicing in eukaryotes. In eukaryotes, a newly transcribed RNA molecule, known as pre-mRNA, is not immediately ready for translation. It must undergo several processing steps to become mature mRNA suitable for protein synthesis. These processing steps include addition of a 5' cap to the beginning of the RNA, known as the 5' cap, addition of a poly-A tail, a sequence of adenine nucleotides, to the 3' end of the RNA, removal of introns through splicing and joining of the remaining exons. The result of these modifications is a mature mRNA. This mature mRNA is then transported out of the nucleus to be translated into a protein. Alternative splicing in prokaryotes. In prokaryotes, mRNA does not undergo splicing of introns or joining of exons, unlike eukaryotic mRNA. However, splicing can occur in tRNA, transfer RNA molecules, to produce their functional form. This involves the attachment of a formalin derivative during the final step of translation. In this step, the processed tRNA binds to mRNA along with ribosomes to facilitate protein synthesis. Examples of alternative splicing. Alternative splicing is involved in every stage of cancer development. Dsam gene, Drosophila, Down syndrome cell adhesion molecule. This gene can produce up to 38,016 isoforms from 95 variable exons through alternative splicing. MHC gene, myosin heavy chain. This gene encodes a protein crucial for muscle cell function. In Drosophila, the MHC gene is a classic example of complex alternative splicing. It contains 30 exons, of which 17 are alternatively spliced. Except for exon 18, represented by a single alternative exon, the other alternatively spliced exons are organized into 5 separate clusters, each containing 2 to 5 exons. Neurexin gene. Neurexins are presynaptic cell adhesion molecules, vital for synapse formation and synaptic transmission. Alternative splicing of Neurexin transcripts produces thousands of isoforms, contributing to the functional complexity of neuronal networks. Importance of alternative splicing. The primary function of alternative splicing is to increase the diversity of mRNA expressed from a single genome, enabling the production of a wide variety of proteins. It helps explain how numerous distinct proteins can be generated from a single gene. It allows exonic regulatory sequences to maintain a wide range of sequence flexibility without affecting coding functions. Small changes in the concentration of regulatory proteins can influence protein interactions, leading to different patterns of exon usage. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!